Chemical digestion begins in the oral cavity. Presence of food triggers release of mucin from the salivary glands through ducts in the mouth. The food is chewed in the mouth where mechanical digestion also takes place. The chewed food or bolus is pushed to the back of the oral cavity by the tongue towards the epiglottis. This flap covers the trachea whenever food is swallowed. The food is then passed to the pharynx, which opens to the esophagus. A smooth muscle that contracts to open a tube more is the esophageal sphincter. Food is guided through involuntary contractions by way of peristalsis. From the esophagus, food particles move down to the stomach, where gastric juice is produced. Gastric juice has a high concentration of hydrochloric acid, causing it to have a low pH. Pepsin breaks down proteins into smaller polypeptides. To prevent breakdown of the stomach wall, pepsin is secreted in an inactive form or as a zymogen of pepsinogen. Food contents in the stomach are churned every 20 seconds to create chyme. The posterior end of the stomach has an opening to the small intestine that is closed off by the pyloric sphincter. Chyme enters the small intestine where most digestion and absorption occurs. Acid chyme passes through the immediate portion of the small intestine called the duodenum. Here it mixes with digestive juices from the pancreas, liver, and gallbladder. In the pancreas, sodium bicarbonate is produced and pH is restored to higher levels. The liver plays an important role in the production of bile, which aids in the process of digestion. The gallbladder stores the bile until it needs to be used. Bile salts allow for fats to be emulsified and for lipase in water to break down fat into the glycerol and fatty acids. Protein digestion continues in the lumen. Larger polypeptides such as trypsin are broken into smaller polypeptides. These smaller polypeptides are broken into amino acids. In the epithelium of the small intestine, villi and microvilli increase surface area that allows for increase in nutrient absorption. The cecum in the small intestine breaks down cellulose. Brush-border cells break down smaller polypeptides into amino acids. Nucleases break down DNA or RNA into nucleotides. Carbohydrates are broken down in the lumen. Polysaccharides are broken into maltose or disaccharides, such as pancreatic amylase. These are broken down into disaccharides and monosaccharides in the brush-border cells. The food then moves into the large intestine where waste is concentrated and water is balanced in the blood through osmoregulation. No further digestion occurs here. The rectum is the terminal portion of the colon and waste is excreted through the